Hello everyone, today we will learn about single and paired end sequencing libraries. This is an example of a double-stranded DNA sequence. Paired end sequencing technology sequences both ends of a longer DNA fragment. In this example, I am showing that the read one sequenced the left side of the DNA fragment and read 2 sequenced the right side of the DNA fragment. Paired end sequencing allows us to determine the two ends of a DNA fragment. This provides certain advantages during some downstream bioinformatic analysis, such as correctly aligning the reads to the reference genome or during the de novo assembly. The distance between read 1 and read 2 is known as insert size. Insert size of a paired end sequencing library is known, or the insert size can be estimated using bioinformatic tools. The information about insert size is often very useful in certain analysis. Now we have a general idea about paired end sequencing technology. So what is the single end sequencing technology? In the single end sequencing, only one of the two reads of a DNA fragment is sequenced. In other words, only one end of the DNA fragment is sequenced in a single end library. There are some advantages and disadvantages using paired end or single end libraries. I would encourage you to explore further about those similarities and differences. In this example, I am showing two FASTQ files that contains raw sequencing information of read 1 and read 2 of a paired end sequencing library. Most commonly, for the read 1 FASTQ file, the naming is similar to this, that has _R1, and for the read 2 it has the _R2. In some cases there could be only _1 or _2. Now I will take a look into those two files. Let's take a look at the first entry. From the R1 file and R2 file, we can see the read ID is identical between these two files. Only difference is /1 and here is /2. So that tells us these two reads in two different files are coming from the same DNA fragment, whereas the left file is for the read 1 and the right file is for the read 2. Now, if we look into the second entry, it also has the identical ID between R1 and R2 FASTQ file. The difference is /1 and /2. These are properly paired reads. These are coming from the same DNA fragment. Also, it is true for the third and rest of the entries. To summarize, the paired end sequencing library provides two raw FASTQ files. One is R1 FASTQ file, another one is R2 FASTQ file. Both the files have identical number of entries as well as they are sorted by the same read ID. Also, I want to mention that in the single end sequencing library, only the R1 FASTQ file is provided as a raw output. Thank you for watching this video. Please check out my website for other courses on genomics and bioinformatics. See you in the next one.